If nature had an instruction manual, it would not look like a clean, organized book. It would look more like a document that has been edited, rewritten, patched, and commented on by hundreds of different hands over billions of years. Modern evolutionary biology has made one thing very clear: life is not built as a simple ladder of progress. Instead, it behaves like a constantly branching, merging, and recombining system of biological information. Genes are not just inherited from parent to offspring in a straight line. They can be duplicated, repurposed, transferred between unrelated organisms, and even originate from ancient viruses. In evolutionary genomics, this is not speculation anymore. It is measurable, sequenced, and repeatedly observed. So instead of thinking of evolution as a designer, it might be more accurate to think of it as an extremely chaotic engineer under infinite time pressure. And today, we are going to look at 6 biological systems that make that chaos visible. The platypus is often the first example scientists bring up when discussing evolutionary oddities. But calling it an oddity is actually misleading, because what it really represents is not randomness, but layered biological integration. It has a duck-like bill, a beaver-like body, and an otter-like swimming behavior. It lays eggs like reptiles, yet produces milk like mammals, without even having nipples. That alone would already place it outside most intuitive biological expectations. But the real insight came from genomics. A landmark study published in Nature in 2008 by Warren and colleagues revealed that the platypus genome is not organized like a clean mammalian blueprint. Instead, it is a mosaic containing mammalian genes, reptile-like egg yolk genes, and venom-associated gene families rarely seen in mammals. In other words, evolution did not erase its past. It preserved multiple layers of it. It stacked them. Even its sensory system reflects this layered architecture. In studies conducted in the late 1980s, researchers like Gregory recorded neural activity in the platypus's trigeminal nerve system. They discovered something unusual for any mammal: electroreception. Around 40,000 specialized receptors in its bill can detect tiny electrical signals generated by muscle movement in prey. This means that in murky water or complete darkness, the platypus is not blind in any practical sense. It is navigating using an entirely different sensory modality. And that is what makes it scientifically important. It is not a broken version of a mammal. It is a fully functional hybrid system, built from multiple evolutionary layers. The green sea slug Elysia chlorotica challenges one of the most fundamental assumptions in animal biology, that animals must obtain energy by consuming organic matter. Instead of fully digesting algae, this slug performs something far more unusual. It selectively retains chloroplasts from algae inside its own cells. These chloroplasts remain functional for extended periods of time, sometimes weeks or even months. This process is known as kleptoplasty. However, there is a biological complication. Chloroplasts are not self-sufficient organelles. They normally depend on nuclear genes from the plant to maintain their function. So the key question becomes, how does the slug keep them alive? A 2008 study published in PNAS by Rumfo and colleagues suggested a striking possibility. They reported evidence that algal genes such as PSPO might be present in the slug genome. If true, this would imply horizontal gene transfer from algae to an animal. Such a mechanism would allow the slug to actively maintain chloroplast function over long periods. For a time, this interpretation was widely discussed and even celebrated as a rare case of cross-kingdom gene transfer. However, later research introduced significant revisions. A 2013 study by Bhattacharya and colleagues, followed by more advanced genomic analysis published in eLife in 2021, failed to find stable evidence of such gene integration. Instead, the more widely accepted explanation today is physiological rather than genetic. The slug appears to maintain chloroplasts through cellular stabilization mechanisms that prevent degradation. In other words, it does not become a plant. It simply keeps stolen biological machinery running for far longer than expected. This makes it one of the most extreme known cases of biological borrowing without genetic assimilation. One of the most surprising discoveries in modern genetics is that a significant portion of mammalian biology is built from ancient viral material. Approximately 8% of the human genome consists of endogenous retroviral sequences. Most of these sequences are inactive remnants, but some have been repurposed for essential biological functions. One of the most important examples is syncytin 1. This protein plays a central role in placenta formation by enabling controlled fusion of maternal and fetal cells. Without this mechanism, mammalian pregnancy as we know it would not be possible. A landmark study published in Nature in 2000 by Mai and colleagues demonstrated that syncytin originated from an ancient retroviral envelope gene. Instead of being eliminated during evolution, this viral gene was integrated into mammalian physiology. Even more remarkably, later studies in PNAS 2005, Dupressoir et al. Showed that similar events occurred independently in different mammalian lineages. This indicates that viral sequences are not just evolutionary noise. They can become functional components of complex biological systems. The Hoatzin is one of the only known birds that relies heavily on microbial fermentation to digest plant material. This system was first described in detail in a 1989 science study by Grajal and colleagues. They found that the Hoatzin's enlarged crop functions as a fermentation chamber. Inside this chamber, dense microbial communities break down cellulose, rich leaves into usable nutrients. This is functionally similar to the digestive system of ruminant mammals, such as cows. However, this adaptation comes with a significant biological trade-off. Because the crop is enlarged, it reduces space for flight muscles and affects aerodynamics. This is one reason why Hoatzins are relatively weak flyers compared to other birds. In addition, juvenile Hoatzins possess functional claws on their wing digits. These claws are used for climbing branches, when escaping predators, or falling into water. This trait is often considered a retained ancestral characteristic rather than a new adaptation. Microbiome research published in 2008 further confirmed that their gut microbial composition closely resembles that of cows. This makes the Hoatzin a rare example of a bird whose digestive strategy is fundamentally shaped by microbial symbiosis. Welwitschia mirabilis survives in the Namib Desert, one of the driest environments on Earth. Despite extreme conditions, it can live for over 1,000 to 2,000 years. A 2021 genome study published in Nature Communications revealed that it experienced whole genome duplication approximately 86 million years ago. In most organisms, such duplication would create severe genomic instability. However, Welwitschia appears to have resolved this through an unusually strong epigenetic regulatory system. DNA methylation plays a central role by silencing large portions of non-essential genetic elements. This prevents unnecessary energy expenditure while preserving genetic complexity. From an evolutionary perspective, this is not simplification, but controlled complexity. It is a system that maintains a large genetic archive while actively regulating what is expressed. the star-nosed mole lives underground in complete darkness. Vision is essentially useless in this environment. Instead, evolution expanded another sensory system to extreme levels. It developed 22 highly specialized nasal appendages covered in mechanoreceptors. A 1995 Nature study by Catania and Kaz revealed extreme cortical magnification in its brain. This means that large portions of its brain are dedicated to processing touch from a very small physical area. Each appendage is mapped with high precision, forming a tactile, spatial map of its environment. One specific appendage acts as a tactile fovea, similar to the visual fovea in human vision. This allows the mole to detect and consume prey in under 200 milliseconds. Essentially, its brain converts touch into a high-speed spatial imaging system. Modern evolutionary biology no longer treats evolution as a purely linear process. Instead, it is understood as a multi-layered system driven by mutation, duplication, symbiosis, and horizontal gene transfer. Across deep time, life has repeatedly reused and reorganized existing biological components. Sometimes viral genes become essential biological tools. Sometimes unrelated organisms merge their functional systems through symbiosis. And sometimes entirely new sensory or metabolic strategies emerge from existing structures. This is why evolution is better described not as a ladder, but as a constantly evolving toolkit. And what we see in these 6 organisms is not exceptions to evolution. They are simply evolution operating without simplification. And that is what makes biology endlessly surprising.